postdoc, and then returned to Vancouver, came up to SFU to start the Human Electrophysiology Lab. Their research is really focused on attention and also what gets us distracted and moves our attention away, which I'm sure he'll talk about today. But he is best known in the Department of Psychology for his sense of humor. Welcome, John.
Thank you. It's great to be here. Thank you for inviting me to speak. This will be maybe light on neuroscience and a little heavy on cognitive psychology. That's what we've typically been doing, but I wanted to introduce myself to the gang, let you know what we've been doing in our lab. I think we could probably borrow some expertise from people with signal processing and that kind of thing. And let me see. Okay. Special thanks to everyone past and present from our lab. There's only 3 previous members on the lab here because I'm going to be talking about some of their stuff today in particular. So they're over there on the left, and Jennifer was just here, stopped in to say hello, so it was nice to see Jennifer. The middle and the right gang are the current gang, and they're wonderful. Thanks to them for doing the lion's share of the work and collecting the data, analyzing the data. And so I'm glad to see Vince on Zoom because I'm going to start off trying to convince you that attention is important, and I'm going to be using a demonstration of Vince's 4-dot masking effect. So the task here is to identify, just name. You don't have to explicitly if you don't want to, but the task is to figure out what shape is going to appear inside the 4 dots. Should be very easy. The 4 dots tell you which is the relevant object. They stay on a little bit longer than everything else, so they just kind of provide that landmark for you to latch onto. I don't know if you can see, there's a tiny shaded square on the screen. That's where the items are going to appear. There's going to be variable number of items over a few demonstrations. So let me see if you can get this first one.
Triangle.
Triangle. Point it up or down?
Down.
Okay, so you can actually get the information about the triangular and the orientation of that. Okay, let's keep going. A little harder. So there was the target along with a distractor, and they're competing for your awareness. So it's a little harder, but not as hard as it'll get.
It's a square.
Yeah, that was Vince. Might have been a square. I didn't see it. And final. Here's the easy one. Okay. So that last one had 16 items. You're probably at chance at guessing what it was. So a couple of starting points here. The fact that the continuing 4 dots masks your perception of the shape inside of it, sometimes, not others, tells us that perception takes time. It's not an instantaneous process, so we, psychology and neuroscience people, like to figure out what's going on in that perceptual process. It was a lot easier when there was 1 or 2 items there. And our capacity for perception and for visual spatial working memory is about 3 items or so. So beyond that, then we're really starting to get the competition from those non-targets. Now I want to try to convince you, hopefully, that we can take that 16 item display and make it possible to do by telling you where to orient your attention in advance. This is a weird room. We'll see if it actually works from your vantage point. But basically, you're going to see, first of all, split second beforehand, a red X. It is going to be at the location of the target. It's not a misguidance of attention. And see if you can lock onto that location and get the identity that time, or this time. Here we go. square. Here's one more. It usually takes one history effect to learn how to do that. Right. Okay, so at least some people were able to get that from their vantage point. So the take-home point here, thanks to Vince, who did this work, is that attention biases your processing towards some items, like 4 dots, or away from other items, to influence what you're perceiving. So I'm going to leave the masking now, but we're going to take these types of attentional paradigms, in this case, a cueing paradigm. And in my lab, what we do is we record EEG while people are doing these types of experiments, and we look at activity that's time locked to different events. So in this case, we might look in particular at what's happening immediately after you get that attentional cue while you're orienting your attention to its location in preparation for the target, and then the eventual consequences of how that attentional orienting influences your actual perception. And then we try to correlate the behavior, the improvement in perception or the decrement in perception with what we're getting with the neurophysiological measures. Okay, so that's the demo. Hopefully you can at least entertain the notion that attention is important. I do have a little bit of an EEG primer. I invited some students and stuff, so I'm actually going to be speaking a little bit at a lower level to show you actually what it is and how we're measuring EEG. I want to focus on our particular ERP approach, or event-related potential approach. We like to try as much as possible to isolate activities of interest that might be linked to fundamental neurocognitive operations, and then I'll take you through a couple of examples of studies. Okay, so I did not pay for this image. You can see that it's still watermarked, but I liked it because it shows you a rat's nest of wires from the EEG electrodes into some electrode interface box, and that will go to some amplifier, which I've put in there, and then that goes to your display. This particular amp is in my lab right now for testing purposes. Tom Spallett has one in his lab as well. So it is kind of the common system right now. Okay, so if we take a system like that using conventional passive electrodes, and we are in some environment that has electrical power sources from the lights, from the wall supplies, we run the risk of picking up 60 hertz noise here in North America. So we can see big 60 hertz sinusoids in our EEG traces, and we can see in the time frequency spectrum a big spike in 60 hertz. So I know that there's at least 2 or 3 guitarists, although Lawrence hasn't been playing so much, he says. And so I've actually learned a little bit about doing good EEG recording from simple technologies that are used on stage here. That's a Canadian-made guitar and a Canadian-made amp. Just thought I'd point that out.
Is that your guitar?
I have not that one, but the same company. It's in North Van.
There's no app.
Oh, you do? Okay, we'll talk later. Or maybe now. Okay. So one of the things people will use on stage is a shielded cable. We've got a signal about 1 volt coming from the guitar, usually stretching 50 feet on a cable to the amp. We don't want that to be hit with lights and other power sources. So the simple solution here is just to shield the inner conductor with some simple shielding connected to ground. That's one thing that we can steal with EEG technology. The other thing is, if you play guitar, you might have one of these on your pedal board. It's called a buffer. It's usually connected to the guitar with a pretty short wire so that the signal from the guitar makes it there. And then the buffer converts what we'll just call the wimpy signal, a high impedance signal, into one that is capable of driving some distances without picking up electrical loads. Okay. So if we now go into the EEG lab, we have a couple of choices from the guitar stage there. We could either shield the electrode wires that you saw, that rat's nest, we could shield all of them, or we could put a pedal board on people's heads and just put buffers very near the EEG electrodes themselves. And then that will be able to drive even a non-shielded cable to the amplifier without picking up much noise. So you might think that's a little strange, having guitar pedals on your head, but these are actually the two common approaches of kind of the more sophisticated EEG amplifiers these days. The one on the system in Tom's lab, and the one that's the most popular, uses what they call active electrodes. They're simply buffers, like I showed you, and they convert the very high impedance signal coming out of your skull into a low impedance signal that is much less immune to picking up external noise sources. The system on the right uses a type of shielding that they like to call active shielding, just because they want to compete with active electrodes. It's a technology called guarding, where the shield itself is connected not to ground, but actually part of your EEG signal is fed onto the shield, and that helps to do a better job at shielding. So both of these types of system will really help you avoid picking up 60 hertz noise. We could also simply use cheap passive electrodes and an unshielded wire, but put people into some Faraday cage, like a sound booth, which is electrically shielded. So just to show you some tests here, we took that ActiChamp system, which usually is used with active electrodes, but you can use passive electrodes. We create our own rat's nest on purpose here. It's a shielded room, but there's lots of power sources in the room. You can see even with passive electrodes, the amps do a good job of filtering out most of the 60 hertz noise. You can see that there was a little spike in 60 hertz, but it wasn't that bad. We use our own passive caps where each electrode costs about $20 each. So they're high quality silver electrode, silver chloride electrodes, but they're pretty cheap because there's no active circuitry on it. Using their active electrodes, we see that we can completely eliminate the electrical noise spike at 60 hertz, but at the cost of $138 per electrode. So if you're using 32 electrodes, you're looking at a cost difference of about $3,500. Some people do 128 channel recordings, so it costs a little bit of money in order to avoid that little spike of 60 hertz. The reason why we don't get too much 60 hertz noise itself, I'll just speed through this a little bit because you guys know about neurons. So here we're recording with one electrode placed on the top of the head, position CZ, and presumably it's over some cortical surface. There's going to be some pyramidal cells in there, and we're picking up the apical side of that open field there. So in this case, I've shown that the open field has a negativity, local field potentials at the top, positivity down below. We don't see anything with respect to the positivity usually, at least in this configuration that I've shown, but we'll pick up that negativity and we'll feed it into the amplifier. But we're measuring an EEG or voltage difference between two electrodes. So we can take another electrode, use it as a reference. We use ones behind the ear, located on mastoid bone. We feed that to the same EEG channel, and that amplifier will amplify the difference between those, the voltages measured at that two locations. So I've shown that the difference there is 30 microvolts. And the reason why we use those differential amplifiers for EEG is because if we do pick up noise, we hope that the noise that is picked up is the same on both the active input and the reference input. And then these amps will do a pretty good job of subtracting that common activity out. And so that gives a picture. There is a ground electrode that's not in that picture, but whenever you see a textbook picture of EEG, they're usually showing one electrode, but we're picking up EEG with two electrodes always. I'm going to return to this idea of the common reference a little bit later. Okay, so that's the EEG primer. Now we're going to get into our own how we use EEG to look at event-related activity. And so again, we're going to be picking up events like that cue and the search array, or in this case, the multi-object array. And for the most part, and what I'm going to talk about here today, we're looking at event-related potentials that are time-locked, tightly time-locked to whatever event that we're looking at. This isn't a great picture of what's actually going on, but the red line shows that we're averaging across multiple trials, and what remains in the average is what is tightly timed. So there are different approaches, and we can do some time frequency analyses that picks up activity that is related to the event, but not tightly timed. But for today's purposes, we're going to stick to ERPs. Okay, so, and I'm going to show you our approach with respect to visual search studies. In each of these side-by-side pictures, you see that there's the identical visual search display. There's 4 identical green circles, non-targets, and then there's 2 different items. One red circle, we'll call that a color singleton or a color oddball. And then there's a shape singleton as well. And we can give different instructions to ask people to tell us something by means of different button presses, something about the circle. So we assume that they're attending to the circle in order to make the response. Or in another case, tell us something about the diagonal. We've developed this technique where we're presenting one of those 2 oddballs on the vertical meridian, above or below fixation, and one of them at a lateral location. We have other configurations as well, but these are the ones that we're actually particularly interested in. And I should mention, I developed that technique with Vince DeLolo and a student named Clayton Higgy, who wasn't on the slide, but Clayton was involved in this as well. Okay, so over the back of the head there, I've got a couple of different cartoon colors. In one case, a blue, and that's to represent a ERP negativity, and red to represent an ERP positivity. So when you're attending, when we ask people to respond based on the color singleton, that item is lateral, and we start to see lateralized differences between activity picked up at the contralateral electrode and the ipsilateral electrode. So I've hand-drawn these waveforms, that's why they look weird. But they're basically supposed to be overlapping everywhere except for in this 100 or 200 millisecond time range. And on the top picture here, what we see is the contralateral solid waveform is more negative than the dashed ipsilateral waveform. Steve Luck and Steve Hilliard First reported that in 1994. It's very popular now. They call it the posterior contralateral N2. No one calls it that. They call it the N2PC. And it's used widely to figure out whether people are attending to a certain item or not. You can do this with people who have spider phobias, and you can show them a display that contains a spider, or social phobias, and you have faces, you have angry faces, happy faces. See whether people are attending to it or not. If we ask people to attend to the special singleton that's not at a lateral location, we assume that they're doing that, but we don't see the N2PC to it because the stimulus is not lateralized. So attentional processing of something that's right on the vertical meridian involves both left and right visual cortical hemispheres. And so you don't get the lateralization unless it's a word or something, and then you'll start to see a left lateralization. Okay, so that little trick allows us to present two things, have them attend to the one on the vertical meridian, and see what's going on, how they're processing the distractor that they're not responding to, so we can't learn about it by probing their responses. And it turns out we can look at their ERPs, and it gives us the exact opposite of that contralateral negativity. So when you're not attending to something salient to the left or to the right, and you're not looking at it, then we see something that we call the distractor positivity, or the PD. So it's the same display, but the ERPs that we get depend on how people are interacting with the display. If you attend to the lateral item, you get a contralateral negativity. If you don't attend to that lateral item, you get a contralateral positivity. And these now have been used quite regularly. I just got today the index of the current issue of attention, perception, and psychophysics, and I see like distraction, attention, visual search, distraction. It's what people are doing these days, and if they're recording ERPs, they're looking a lot at these two components. Okay, so now if we look at this, the difference is the component of interest. That's what we're interested in, but it's riding on top of a lot of other activities. And if we try to look at the difference in, let's say, a topographical map or a source analysis, it's hard to do because what we see is the P1 and the N1 and the later P3, and the difference is a small thing. So what we try to do is we try to pull out the difference and subtract as much of the other stuff out as possible. So I'll show you an example, not of N2PC. This is a third component that we discovered a few years ago now. And so we had what we called a singleton detection task. There's 16 items on the display. All of them are identical. They could be vertical, like you see on the bottom here, or they could all be horizontal. So you don't know if you're going to see all vertical or horizontal. That would be the singleton absent display, and half of them, they have one item that's rotated 90 degrees, and so that's a singleton detection, a singleton present display. They're asked to hit the button as fast as possible to tell us whether the singleton is present or absent. Simple task. So the singleton absent display, no one previously has really cared about that, but we can use that as our baseline, and we can subtract away activity that's elicited by the singleton absent display from singleton present displays. The difference we would ascribe to the processing of that singleton. Okay. So in A there we see present and absent displays. The absent is the dotted line. You see that the initial uppies and downies, the initial positive peak, P1 and N1, they're all the same. And the difference is a little bit later on. The singleton absent elicits a bit of a positivity, but it's not as much as the singleton present displays. So if we just move over to the right there, we see the present minus absent difference waves. And That's a nice flat line with a big hump, and then we can study that hump without the rest of the stuff going on. And so, for example, we can see that the hump starts earlier. We call this the singleton detection positivity. You might just call it an occipital P3 if you've heard of the P3 component. We see that it starts a little early when there's more items in the display because it's easier to pick the gradient discontinuity out when there's a larger backdrop. We see that when people are fast, this thing happens early. We see that when they're responding more slowly, it happens a little bit later. So that allows us, this subtraction method allows us to study the activity and look for small differences. And the topo maps show what it looks like with pre-subtraction and post-subtraction. So in both cases, singleton present and absent displays have this positivity of P3B component, maximal kind of at the midline parietal scalp. But when we subtract one from the other, all of that P3 goes away, and we're left with bilateral occipital activity. And we can source analyze that activity. We can model it quite well with just two dipolar sources in the occipital cortex, and that's very easy to study. So the subtraction helps us. The prior slide only showed you what's happening on the ipsilateral because it's a nice round positive hump. Here in panel B, we're showing ipsilateral and contralateral waveforms. The contralateral one's a little bit more complex, and the difference between the ipsi and the contra is that N2PC component. So here, I'll go back a couple of slides, or maybe, a few slides. Here we see just the N2PC, but it's riding on top of a lot of things. We can simplify it down to this. We still can't isolate the N2PC separate from the singleton detection positivity, but we could if we just subtracted the baseline or the contra from the ipsi or the ipsi from the contra. And that will give us another subtraction. I'm going to give you one more method that we're starting to use to do some ERP isolation, in this case of N2PC type activity. So again, here's in A, you see the typical visual evoked potential, P1s, N1s, late positivity, and the N2PCs riding on top of all of that. We can do the singleton present minus absent. P1s and N1s go away, so that's just what I've shown you previously. Now I'll just mention what the common reference was. So every electrode on the head, including P07 and P08, where these waveforms come from, were referred to averaged mastoids. So we do this digitally after the fact. We average the mastoids. Everything is referred to the average mastoids. Now they look close in, physically close, but there's not much going on at the mastoids because the bone is very thick there. So we got lots of activity at P07 and P08, even though they're close to the reference electrode. Remember, these are differences. So P07 minus the mastoid. So it's a pretty big difference. We get lots of activity. We've started to play around with the idea that we can instead use what I'm calling a local common reference. So we'll digitally re-reference this to something in between the contra and ipsi electrodes. So here I've shown you in the middle is an electrode OZ. So we could use OZ as our reference. And you can see that the P1 and N1, they don't go away entirely, but they get a lot smaller because the P1 and N1 are also picked up at OZ. So now because of our differential amplifiers, some of that activity is subtracted out. Now the nice thing is once we do that and then we subtract singleton absent ERPs from singleton present, we essentially get a flat line with a difference, and we can start to blow up that difference and study it a little bit more. So this is just an example of how we isolate activity. If you compare the diagonals in A and the bottom of B, you'll see that there's a lot less going on. We can subtract out a lot of activity either by creating difference waves and by using this weird local common reference that people usually don't do. Questions thus far? Okay, so now I'm going to take you through a couple of studies. We use all of these things. I'm not going to talk, I don't think, about the distractor positivity, but we will, I think, focus on some N2PCs. I should say that when people do attention cueing studies, the demo involved cueing, where we use that X, the red X, to get our attention to a location in advance. So we'll call that attention cueing. And there are many experiments where people use what we'll call invalid cues. So they're cues that pull you to one location, but the target's not there, it's over here. Okay, so there is a cost associated with those types of invalid cues. You're slower to respond to the target on those cases. So why is that? What causes that slowdown? I'm going to try to convince you that what people think causes the slowdown is not what causes it. That's the purpose of this. This actually was an honors project in my lab. So it was quite nice to do this with an undergraduate honors student. Okay, so this is an example. We're not starting off with the most simple attention cueing experiment, like just presenting a red X. This was a cueing experiment that was used by Chip Folk and colleagues to study what they called the contingent capture of attention. What is attention capture contingent upon? According to Folk and colleagues, it's contingent upon their top-down attentional control settings, or their attentional set. So here, in this example, the participant is in the second display, looking for an X that appears with a black X, appearing with some black pluses. So their attentional set is probably for some sort of form feature, right? Because the target or could be size, the way I've drawn it, I think, but the target doesn't differ in color, but it does differ from the rest of the non-targets in terms of its shape. So in that type of situation, we know that nothing in this other irrelevant display is going to capture attention. People won't inadvertently attend to any one single colored disk because A, they're not salient, there's not one that's more salient than the other, and B, nothing looks like the target, nothing resembles the target. So that would be called a mismatch distractor display. Nothing matches the target. So attention doesn't go anywhere when you present that distractor display for, say, 100 milliseconds. And then there's a couple 100 milliseconds delay before the target display happens. They're to search for the target. Then it was Mark Neimer's group that first looked at this with N2PC, found that, in this case, the target elicits N2PC. So now if we just change the target to a red plus sign, now it differs in color and only color. So that attentional control setting for color, or in particular for red, will make you, turns out, it'll make people attend to the red distractor as well. And so that was first published by Mark Neimer's group. So now I want to kind of underscore what the field's interpretation is on this type of trial where the cue is bottom left and the target is somewhere else. Okay. So those trials, we know people are slower, and this is the reason why people think they're slower. Okay, first, we know that attention goes to the distractor location. We can measure N2PC to it to confirm that it was selected preferentially. Then, this display doesn't last this long. It's usually 100 milliseconds. And then quickly after that, the target display happens. And it's about that quick. So if your attention is pulled over by that bottom red thing, when the search array comes on, you're still attending there, according to this explanation. So you've just selected a cued non-target. You've processed something that's not the target. And the person has to identify it and says, that's not the target, I have to go find the target. And that takes a little bit of time. You'll get an N2PC to the target as well. The the non-target N2PC and the target N2PC kind of bleed together. You can't separate them because they're on the same side in this trial. But that time it takes to realize the cued item is not the target and to shift to the target, that is what people think causes the delay. In our 2022 paper, we called this the serial orienting hypothesis, and we linked it back to Mike Posner and other people who never explicitly said it. So I was happy to find this this year, where basically they spell out our serial orienting hypothesis. So they didn't read our paper. That's clear. So they talk about the delay in responding on these different location trials, here labeled as invalid in the bar graph, invalid cue trials. That these types of matching cues capture attention to the correct location on valid trials, but that attention has to be disengaged from a cued distractor position and redirected to the target on invalid trials. So that's exactly what in 2022 we tried to convince people, like I'll do today, that that's not the case. So people are still going with this textbook explanation. Okay. So how did we study this? Well, first of all, in the demos that I showed you, which were used by us and by others, previously had one item in each quadrant. And that makes it difficult to study. I'm going to go back because if you have an invalid trial where the distractor is on the left and the target's on the right, they're both going to elicit N2PCs, and you're going to get some weird combination of contralateral negativities, contralateral positivities, which can happen. I haven't talked too much about them. And they all mix together. So ultimately, we decided we needed some midline. This was actually Ashley Livingstone's idea. She left after her master's, so she didn't actually run this study. Jennifer Hoffmeister did, but it was Ashley's idea to use our trick of the midline positions. So we just turned everything 90 degrees, and we used these types of displays and other ones, but we always had something on the midline. So let's look through a couple of these ideas here. I'm gonna, and the other thing, notice that I've kind of put these activity distributions as like selection activity. And we used to plot, and we probably still will plot for a while, ERPs with negative up. So I'm now switching to my old way of plotting ERPs with negative up. So N2PCs are upward deflections like these activity distributions. I couldn't change them for this talk. Okay, so let's focus on a couple of these. First of all, in C, this is a situation where the match distractor is at a lateral location here. Well, on what? The blue one. We're going to go with the blue one. They're looking for the blue target. So the blue disk, the distractor, is going to capture attention to a lateral location. Then when the search display happens, they're going to be attending to that cued non-target. That's not the blue item. So they're eventually going to have to shift attention down to this blue item here. Okay. That blue target won't elicit an N2PC, even though it's attended, because it's on the vertical meridian. So this will elicit an N2PC. I've indicated that here. The cued non-target presumably will elicit an N2PC. That's this one. The target will be selected, but I've put that activity distribution in a dashed line because we won't see the N2PC to it. In order to see contralateral negativity, we need a lateral target. Okay, so notice here that relative to things that I've grayed out, that there is a delay in the target N2PC. Okay, it happens after the cued non-target N2PC. So let's look at this other configuration where the target is is at a lateral location, but the distractor is at the midline. So here, early on, the distractor pulls you to the midline. We're not going to see an N2PC to it because it's not lateral. The cued non-target's not lateral, so we're not going to see any N2PC to us. This allows us to isolate the target N2PC now, unlike last time where we were isolating the cued non-target N2PC. So here we see that the target N2PC is there. And now, if we're able to compare, keep in mind what I showed you were both invalid cue trials. The target is not at the cued location. We should be able to look at the isolated target N2PC and compare it to the isolated cued non-target N2PC and see that the target selection is delayed on invalid cue trials. And moreover, it's not just delayed, but people select that cued non-target first. Okay. That would support the serial orienting hypothesis that I read earlier. That's not what happens. Okay. So here I'm going to compare the top and the bottom configurations. The top one, which I haven't mentioned before, is the valid cue trial where the target is the item at the cued location. Okay, so what happens there? It's the red figure here. Again, these are ERPs with negative up. This little gray box is when the distractor display happened for 100 milliseconds and a couple hundred milliseconds of free time before the target search array happens. So this is actually time locked here. Everything is time locked together. So this upward negativity is the N2PC to the match cue. So if they're looking for a red U, by the way, the task was to indicate where the missing edge is on the top or the bottom. Okay. So we get an N2PC to the cue, the red disk, if they're looking for a red U. Then early on, we actually see some positive stuff. After the onset of the search array, we get some positive going stuff that happens contralaterally. So it's labeled CP there because we're kind of lazy now. We just called it the contralateral positivity. And following the contralateral positivity, then we see, and that's to the target, then we see the target N2PC. So contralateral positivity, we think, is in the pre-attentive range because we're saying, or at least pre-selective range. We think that selection is indexed by this N2PC. So there's the N2PC. We can mark its time there. And that's on valid trials. Then the dashed line is the bottom below where it's an invalid trial. They should actually find the target later. But we've isolated it so we don't see any selection of midline distractor or midline cued non-target. So we still get a nice isolated N2PC, flat line here, and then we get the N2PC, and it happens in the exact same time. Okay, it's not delayed at all. So that is interesting because there's no evidence that an invalid misdirection of attention causes you to find the target any later than you otherwise would. Another comparison, it's the same red tracing for the valid cue trial, but now we're bringing in the isolation of the cued non-target N2PC. That was supposed to be an early N2PC because you're looking at the item, or you're attending to the item. But it turns out, we see that again, there's an N2PC to the distractor or the cue. There's this contralateral positivity, which we think is attention turning up the perceptual processing, early perceptual processing of the cued item. And then finally, we get an N2PC to the cued non-target, but it's the one that's delayed. Okay? So people are not getting to the target late, they're actually attending to the cued non-target. If we go back to these kind of pictures, this is what we think is happening. They first attend to the, and in this particular case, they're looking for a red target U, gap on the top or the bottom. They attend to the red distractor because it matches the color of the target. We get a boost of perceptual processing of the cued non-target, in this case the blue one, but it's not yet attended. They lock their attention onto the target that they're looking for, and then only then they lock their attention onto the cued non-target, and from then on, they process the identities of these items for several hundred milliseconds. Okay, so once they find the red thing, and I've learned this from Vince, it's a dual stage process. First step, find the red thing, found it. Okay, is the gap on the top or the bottom? You still have to do that process. So you get an N2PC, and go back, and then you get a second hump, which is related to the identification stage, that second stage. Found the red thing, that's the first hump, the N2PC. Where's the gap? It's on the top. That's what people sometimes call the contralateral delay activity or something like that. It's the second negative hump. So what we think is going on, and we had reaction times to back it up, which I'm not showing here, we think that when you attend to both things concurrently and you're trying to identify the red thing, that since you've pulled out information about the blue thing's identity, it will interfere with your processing, like at the stage of identification. So what does that mean? It means that people are actually only slow on invalid cue trials, on trials which I've shown here, when the cued non-target is not the same as the cued target. Okay? So if you process a gap on the bottom and a gap on the top, now you've got to overcome that incongruity, and you have to pick which one you want to respond to. If they're both gap on top, you're not slow enough. So we don't think that delay on invalid trials is due to some delay in getting your attention to the target. We think it's due to somewhat late congruency issues when both items make it to the second stage of processing. Okay, so why do we think that this is important? There's a lot of models about orienting of attention, about visual search, etc., which assumes that certain areas play certain roles in attention, shifting attention, disengaging, re-engaging, and all of those assumed roles are based on these types of invalid cue effects. And if you have damage to parietal cortex, the cue effect changes in a certain way. But all of their interpretations are based on this idea that we're slow because we have to get attention over there and we're delayed. We also think it matters because most models of attention, like moving your eyes around, assume that we have one unitary focus of attention. So if we want to shift it from here to here, it might even go through space like the eyes do and then lock onto that. But what we found is that you're actually attending items concurrently, and you're pulling out information from two very discrete items at the same time. So if we want to learn about things like attention deficits, how attention might change with aging, with developmental disorders, we should know what's happening in the healthy brains. I'm basically out of time. I want to just end off with a little bit of another study, which gets at this idea of, do salient items capture attention? And we've played around with this with these types of go, no-go tasks. So in this case, attend to this singleton because the whole array is blue. That one we wouldn't attend to. We would tell people, you don't have to respond. This is a no-go trial, but it still contains a singleton. So the question is, does that singleton capture attention? And it turns out we found out that if we tell people not to respond, indicate whether the singleton is present or absent, for yellow displays, we don't get an N2PC to the singleton. So even though it's salient, it doesn't capture attention because it's not behaviorally relevant. Daniel decided that we're going to get rid of the two colors because the go gives us one, the no-go doesn't, but the no-go is not the same display, and maybe there's some reason why that's a problem. So we just got rid of the two colors, we used one, and we cued people to search or not to search. So the time the cue was presented for 100 milliseconds, 8 to 1200 milliseconds later, the search array came on. One of these gigantic symbols meant you should tell us whether the singleton is present or absent. We call that go trial. The other symbol says you don't have to respond on this trial. Again, we get an N2PC on go trials, not on no-go trials. It's the same display. We just tell people you can chill on this trial. You don't have to search. What's also cool is we looked at the ERP to the cue itself. They give some early activities that are similar. Then there's some differences in the P300 time range. They're really looking for the go symbol, and that's more behaviorally relevant. So the P3 is a little bit bigger. And then we get this big difference in late sustained activity. On the one hand, the no-go activity is sustained positively. And on the other hand, the go activity continues to get more negative until the search array happens. And the search array happens randomly sometime after 800 milliseconds, 8 to 1200. So eventually this negativity dies away. Now, there's the difference wave activity. If we take the go minus no go, it looks like that on the head. We're calling it right now a preparatory medial frontal negativity because it's largest over medial frontal scalp, sort of. There's a lot of anticipatory slow waves in these types of S1, S2 paradigms. You might have heard of the CNV, that's the most popular one. That's linked more to responding. This isn't going to be a CNV because they don't know what button to press yet. They don't know if the singleton is going to be present or absent. We looked at this, we looked at go trial activity in particular when they have to switch to a go trial. So they've had a no go trial, they've chilled for 3 seconds, now they get a go trial, and that's the cue activity towards the middle to the end of the cue target time range. If they get a second go trial in a row, there's a little bit different. So we can look at the difference by creating a difference wave, and we see a couple of things, including this negativity over medial frontal cortex. So it takes more effort to switch to a go trial, to respond, to get ready for the first go trial after a break, after a no-go trial, than it does to continue with continued go trials. These activities, the preparatory MFN in general, and the earliest portion of this difference wave at FZ, the mediofrontal negativity that comes on early in this case, actually are predictive of whether or not the target will elicit an NTPC on a go trial. So you select it. I'll just look at here on the left side. If people don't have much preparatory MFN in the Q target interval, they actually have no target N2PC. In fact, it's positive, as if they maybe suppressed it. Similarly, if they have early difference, more activity early on when it's a switch trial, then that also correlates with the target N2PC. And finally, I don't have a slide, but the no-go positivity that I showed here, this activity here on a no-go trial, which we think maybe, this is new, we got this yesterday, and we're still collecting data. We think this might be some sort of inhibitory control. People are saying, no, let's not respond, let's stop. That activity, preparatory activity on a no-go trial, also correlates most strongly with the target N2PC on go trials. So people who are good at inhibiting on a no-go trial are also the best at finding the target on go trials. That's a simple story. I will end there. There's a summary. Thanks to these people again, and thank you for having me here.
Thanks again.
Thank you.
John. Our next two sessions for the Institute seminar series are actually going to be trainee talks, trainees that were lucky enough to get travel awards. So we're going to ask them what it was they presented at these conferences. So that'll be March and April's seminar series we'll have. So what I did on my summer vacation kind of talks. So thank you. Thanks again.